So now, well, I've been tasked to talk about the last leg of the Tara expedition, which is the Polar Circle expedition. And I will, well, both Lee and myself will speak about this expedition on behalf of the Tara Ocean Consortium. I will briefly provide an overview or kind of a context of the expedition, an Arctic context, and Lee will kind of focus on the sampling strategy and also the relevance of the data. So if we compare the Arctic Ocean with all the other oceans that have been visited by Tara, in fact, the first thing we have to kind of acknowledge is that the Arctic Ocean is relatively small. It's only, I would say, less than 1.4% of the global ocean. So that's rather small. And something which is kind of striking as well is that this ocean is encased within land. So it's surrounded by various land masses. There is very big rivers on these land masses, kind of dispensing a huge amount of fresh water into the ocean. There is some small collections, one, well, the largest being to the Atlantic via the Fram Strait, I don't know, yeah, and kind of a smaller one kind of via the Bering Strait to the Pacific, and something which is kind of a very distinctive feature of the Arctic Ocean is that there is ice in there. And in the winter, it's almost entirely covered by sea ice. There is a little bit, or kind of about 1/3, still remaining in the summer in the deeper kind of in the deep ocean, or in the kind of well, in the central province. And something which is also important to mention is that the Arctic is currently experiencing a very strong warming, and you can see all these red colors. In fact, it's kind of the strongest warming on the planet. And it's from 1 to 4 degrees, or up to 4 degrees in some region since the last 50 to 60 years. And this warming, and that is well known, in fact, is kind of provoking a dramatic decline in the sea ice extent in the Arctic. And here what you can see is the summer sea ice, so the lowest amount of sea ice in the Arctic. And you can see that it has been gradually decreasing over the last 30 years, from kind of about 8 million square kilometers in the late 70s to kind of between, I would say, 4 and 5 million kilometers now. And in fact, this big decline in sea ice is inducing dramatic changes in the phytoplankton, or even on the Arctic biology. So if we take primary production, all these red colors that you see around in the Arctic are an increase of the production. That was between 10 years ago and kind of nowadays. And what's also important to mention is these blue patches here and here, which in fact are decrease in the primary production. So changes in the sea ice, or environmental changes, are kind of inducing changes in the quantities of, I would say, the food available to all the organisms in the Arctic. And it's not only the quantity, but with the decline of sea ice, you have an increase in time of the productive period. And whilst in the past we were having only one bloom, now we start to see some autumn bloom appearing. And you can see that all these red colors is kind of the percentage occurrence, or percentage change in the occurrence of a double bloom. So not only a change in the quantity, but also in the timing of the primary production. And a third change, in fact, is in the quality of the food. So Bill Lee and collaborators, they found that big phytoplankton cells were actually kind of, well, I wouldn't say stable, but decreasing a little bit. And in fact, what was taking their place is much smaller cells. And Lee will show some results about that later on, but there is changes also in the quality of the food. And well, phytoplankton is the first step. They are feeding all kind of the kind of higher trophic levels. And the traditional vision is that basically there is, in the Arctic, there is a strong connection between the upper levels and the benthic, the benthos, or the bottom of the ocean. And people already start to see, like Jackie Graham-Meyer in the Bering Sea, that we are now switching from a benthic-dominated ecosystem towards some much more planktonic-dominated ecosystem. So major changes. And, well, if you think about the benthos, it is impacting benthic fish, marine mammals, and also diving birds, which are feeding on benthic invertebrates. So I would say that's why expeditions like the Tara Arctic or the Polar Circle expedition are important. All the data that I showed, apart from the data from the kind of the NASA instruments, are made from very regional studies. And, well, for I would say, the first time, the Tara expedition was really the opportunity to get around the Arctic kind of a data set. So that was very important, and that's why the lab, our lab, has been involved, in fact. And as you can see, this is the track of the Tara across the Arctic, and you can see that they sampled in areas which were kind of heavily influenced by the Atlantic Ocean, so quite productive areas and kind of warm areas, and then kind of they gently visited some kind of more Arctic regions, I would say, and then went down all the way to the Northwest Passage and back to visit us in Quebec City. So I'll stop here and up to Lee.
Okay. So I just want to share some work in the pipeline that we are doing with respect to the Arctic expedition. And just the fact that Tara was able to safely complete the circumnavigation through the Arctic Ocean is an accomplishment by itself and the testimony to the changing Arctic. But in terms of science, there were also some significant accomplishments. Emmanuel was talking on the underway sampling. We were able to complete underway measurements along the 27,000 kilometers of the navigation through the Arctic Ocean, measuring the properties of seawater like temperature, salinity, pH, and partial pressure of carbon dioxide, and also the properties of particulate and dissolved material in the water using state-of-the-art physical, optical, and imaging sensors. And in these maps, you can see, where's the laser? Here. So the top map shows the surface salinity measured by through the underway system, and the bottom one is the temperature. And what you can see here clearly is you see the influence of the Atlantic water, the relatively salty. The red color is the saltier, blue color is less salty, saltier and warmer water coming from the Atlantic through the Bering Sea, and then the fresher, colder water characteristics of the Western Arctic. And on this figure, this is the biomass of phytoplankton estimated from the chlorophyll along the track of Tara during the circumnavigation. But this is only the surface, and in addition to this underway sampling, we also accomplished measurements in 50 additional stations where we took samples from environmental data and characterizations of plankton using both vertical profiling system, collecting discrete water samples for chemical analysis and particle characterization, and in sub-sample of these stations, we also took whole water samples for size fractionated plankton analysis for the metagenomic analysis. the same way that was using the same methods that were used in Tara Ocean. And this is just a sample of the distribution of stations in the Arctic Ocean, showing the vertical, now looking at chlorophyll, the proxy for phytoplankton biomass, on a vertical scale. So the top of each bar is the surface going down in the water column. And it's important to highlight here that in the Arctic, a lot of the biological activity is subsurface. So just looking at surface samples is not enough. We have also to look at depth. And I want to mention that sampling, I think the previous talks have highlighted the challenges of sampling all the diverse measurements that our oceans took. Doing it in the Arctic, it's even more complicated. And without these two people, Marc Picheral and Claudie Marek and Céline Demier, I would also add, this work would not have been possible. In addition to these samples, we also, the sampling program including parameters that are directly related to anthropogenic effect, including plastic and mercury. And this is a sample that we took, this is just a photo that I took when we brought a sample up from the Arctic, from the Kara Sea at 77 degrees north. You think you are at the end of the world, right? And away from any human influence. And those arrows point to little small plastic fragments that were in the sample. Same for mercury. Arctic marine mammals have concentrations of mercury, one of the highest in the world. And that, of course, have a very important effect on local communities that feed mammals, marine mammals, and are important part of their diet. So all these samples are now, most of these samples have been processed and now are in different stages of validation and analysis. And I just want to share one direction that we take with these samples, looking at phytoplankton community structure. So the traditional view of the pelagic environment in the Arctic is that of a low diversity and very simple food web, mostly dominated by few large species. But current study in the Arctic start to challenge this view, show that diversity might be, especially of that of protists, is possibly higher than what we saw before, but also that small phytoplankton play an important role in the Arctic. But all these studies have been done on a very local scale. Now we have an opportunity to look at those structure of planktonic ecosystems on a pan-Arctic scale, and this is really the novelty of this expedition. So in the underway system, we had an imaging flow cytoboat where we continuously took samples of phytoplankton. And I just want to show you, this is a combination of images collected by the imaging flow cytoboat, just to highlight the diversity of protists in the Arctic. This protist includes both heterotrophs and autotrophs, but one of the dominant phytoplankton species, one of the dominant diatom in the Arctic is Chaetosaurus. Along our samples, we identify at least 19 different species of Chaetosaurus. So the flow cytoboat we collected more than 2 million images. 40,000 of them have been manually identified. So now we have a good training set and a catalog of phytoplankton of the Arctic. And what we do is we use this training set to use computer-assisted sorting of all those images that we collected into taxonomic and functional groups so we can start and understand and create the structure of the ecosystem. We combine these images with images that were collected by another imaging particle counter, a FlowCam. The FlowCam, we used it to look at discrete samples, so the flow cytoboat provides the underway measurements, and the FlowCam gives us the information on the vertical dimension. So this is the larger phytoplankton. These are what people have mostly looked in the Arctic. But recent studies show that the small ones are actually very, very important. So we also used flow cytometry to look at the pan-Arctic distribution of the nanophytoplankton and the picophytoplankton. And so this is going from the Barents Sea, Atlantic influence, into the Western Arctic. The top one is data from flow cytometry looking at the biovolume of nanophytoplankton. These are the blue ones, and the light blue are the picophytoplankton. And what you can see is that while nanophytoplankton dominated the Russian basin, picophytoplankton dominated in the Western Arctic. And when we looked at pigment, look at size fraction of phytoplankton based on pigment, we see a similar trend, a relatively dominance of the nanophytoplankton in the Eastern Arctic and dominance of the picophytoplankton in the Western Arctic. The next step is to combine this imaging and flow cytometry with the genomic data to really tell us not only about morphological diversity, but also about the genetic diversity that you have heard before. So here we have heard all the talks about the genetic diversity. Now we are trying to integrate it with also morphological diversity. And I want to end with the last paper, with one slide from one of the 5 big papers, from Shini's paper. Tar oceans is not just about the Arctic, it's about the global ocean. But what Tar Arctic adds to the global data set, so this is a graph showing the microbial diversity as a function of temperature and latitude. You can see that there is lower richness at high latitude and near the equator, and higher diversity at mid-latitudes. But if you look at the polar regions at the low latitude, we have very few samples. Now we can use the Tar Arctic data and really complete this global view. So just stay tuned. So.